So I'd like to thank all of you for being here today, as well as those of you that are online for this very big day, which is the release of the Tufts Center for the Study of Drug Development's most recent analysis of the cost of drug development. I'm Ken Caton. I'm the director of the Tufts Center for the Study of Drug Development, and I'm a professor here at the Tufts University School of Medicine. And before I introduce the study's principal author, Joe DeMasi, I'd like to just take a minute to say something about who we are, the Tufts Center, and something about the significance of today's findings. The Tufts Center for the Study of Drug Development is a multidisciplinary academic group that's based here at the Tufts University School of Medicine. Since founded in 1976, the group has studied the economic, legal, political, and scientific issues that affect the development and regulation of new pharmaceutical and biopharmaceutical products. And for nearly 40 years, our mission has stayed basically the same, which is to provide strategic information to help all stakeholders improve the efficiency and productivity of pharmaceutical and biopharmaceutical R&D. These stakeholders include not just the developers, but also the regulators and the policymakers, both at the state and the federal level. And that's, of course, very important in areas like Massachusetts, where there's a very vibrant life sciences community, but also payers, academics, contract research providers, and investors who use information that we put out to help make decisions regarding investments. We fulfill our mission through our research, our scholarly publications, our professional development programs, and our educational opportunities here at the university. Tufts CSDD is the leading source of scholarly analyses on the development, regulation, and utilization of new pharmaceutical products. So just a few words about the cost study. On average, the Tufts Center puts out about 30 peer-reviewed publications, reports, and other types of written material over the course of a year. On the other hand, our cost study analyses are typically some of our most cited and influential works. And the reason for this is that the drug development cost figures are critical across a very broad swath of the life sciences enterprise. For example, drug developers use this figure for making decisions about research priorities and resource allocations. Policymakers and regulators use this information to help make decisions about crafting good legislation and regulation to stimulate innovation and ensure that vibrant life science communities remain sustainable and healthy. And of course, investors use the figure to assess opportunities in the life sciences sector and the risks involved in investing in new companies and in new products. So now it gives me great pleasure to introduce my colleague, Dr. Joseph DeMasi. Joe will present the findings of the most recent cost analysis, and what we will do is he will take questions from the audience and online after he's done speaking, and that will go until about 11 o'clock, and then from 11 to 12 he will be available for one-on-one discussions or questions with anybody who would like to speak with him directly. Dr. DeMasi is the Director of Economic Analysis at Tufts CSDD and is an Associate Professor at Tufts University School of Medicine. He's an internationally known and highly respected economist with an extensive list of scholarly publications in peer-reviewed journals that quantify the time, the cost, and the risk of new drug development. So it gives me great pleasure to introduce Dr. Joseph DeMasi.
Good morning. Today I would like to share with you some of the findings we have for our newest study on the costs of new drug development. Since its inception, the Tufts Center for the Study of Drug Development, originally at the University of Rochester, has gathered and analyzed data on the drug development and drug regulatory processes. And it has published these analyses in numerous studies, as Ken has mentioned, to better inform developers, regulators, policymakers, and other researchers of trends in biopharmaceutical innovation and factors that affect that innovation. This study is one that fits squarely in that tradition, and it is, in fact, one of a series of studies dating back to the 1970s. These studies use comparable methodologies, so they permit apples-to-apples comparisons of drug development costs over an extensive period of time. Okay. Now, let me begin by first noting that my co-authors for this study are Henry Grabowski of Duke University and Ron Hansen of the University of Rochester, and Ron is here today. What I intend to do this morning is start with the top-line findings and then work our way methodologically and analytically to get us to that figure. Specifically, I'm going to walk us through the nature of the data, the methodology used to develop an estimate of the pre-tax private sector resource costs of getting new drugs from discovery to the marketplace, and the major results that we obtain. Along the way to getting the top-level cost per approved new drug metric, we can get a lot of results about the components of the drug development process. I'll show you some of those results, which include cash outlays by general development phase, what I refer to as clinical phase, transition and overall clinical approval success rates, and development times by phase. We also made use of the information available from the survey and other sources to develop an estimate of the post-approval cost per approved new drug, which can be added to our pre-approval cost estimate to get an estimate of the full life cycle R&D cost per approved new drug. I should say that I'm using the term drug in this presentation as a shorthand for both drugs and biologics. Both large and small molecules are included in the cost data set and in other data sets used for the analysis. Now, as I mentioned, we have done a number of studies of this kind with similar methodologies over the years. So I will use past results to indicate the extent to which R&D costs have increased over the decades. Finally, I will then present an analysis of the extent to which various parameters or components of the drug development process have individually contributed to the change in R&D costs relative to those for the previous study in this series. Okay. Now, our top-line estimate is shown here. The cost per new drug approval, inclusive of the costs of drug failures and what we will refer to as the time costs of new drug development, is nearly $2.6 billion. And I'll show how we get to that number. Okay. But first, in the way of a background to this process, I think the simplest way to look at the relationship between drug development expenditures and new drug approvals is to examine published industry data on R&D expenditures and approvals. This can give a general picture, albeit a very limited one, of what has happened over time. I inflation adjusted the R&D expenditure data here, but I should note that these data are only for member firms of the Trade Association Pharma. However, the approvals are for all firms, so the relationship between expenditures and approvals here is actually somewhat understated. It's also the case that there's a substantial lag structure between when R&D expenditures are incurred and when drugs get approved. So this presents a very imperfect picture and can't be used to get specific cost estimates, but given the trends shown, it at least suggests, I think, that R&D costs have been increasing significantly over the decades. Okay, so let's move on to the actual study and the data and methods that were used. We had 10 firms provide data on 106 investigational drugs, some of which have been approved and some of which failed in development. In fact, the vast majority failed at some point during clinical testing. The inclusion criteria for these compounds are that they were developed under the auspices of the survey firm so that all of the cost data were available, and they first entered human testing anywhere in the world from 1995 to 2007. Based on pharmaceutical sales, 5 of the firms are top 10 companies. 2 of the firms were ranked in the 11 to 25 group. Another 2 were in the 26 to 50 group, and 1 was outside of the top 50. Now, some of the cost and time data, let's go back here. Oops. Some of the cost and time data ran to 2013. A small portion of the sample was still active in development. And this will actually lead to a slightly conservative estimate. It won't matter much because it would only affect one phase for each of a handful of drugs. Given high failure rates in development and so relatively few compounds that last to phase 3 or approval, we oversampled drugs that lasted to later development phases so as to get as much information as we could on those phases. So this is what is referred to as a stratified sample. We then weighted the results to reflect the distribution by phase and outcome for the population. Finally, we gathered data on annual company R&D expenditures over several decades, broken down in various ways. This was done primarily to get estimates of the pre-human, that is, discovery and preclinical development costs. Now, this is a list of the various elements of the process that we had to estimate to get an overall cost figure. By out-of-pocket, I mean the actual cash outlays. For the clinical phases, we are including all R&D related costs for the molecule that occur during the testing phase, not just direct trial costs. Clinical approval and clinical phase attrition rates are used to get what are called expected phase costs and to move from cost per investigational drug to cost per approved drug. Expected costs are mean costs weighted by the estimated probability that a randomly selected drug will enter a phase. Development times for the entire process and a cost of capital estimate are needed to get what you can call the time costs of drug development. That is, there is a cost to investing in pharmaceutical R&D years before any potential returns can be earned on that investment. So we will need to estimate a development timeline, spread the out-of-pocket costs over that timeline, and use the cost of capital as an interest rate, if you will, to capitalize those costs forward to a certain point in time. Now, this slide just represents the first level of estimation. We look at the compound level cost data on the investigational drugs in the sample. The data were given by clinical phase in year. As I mentioned, we oversampled compounds that proceeded to late-stage testing to get enough information on those later stages. And then we weighted the data to reflect the population distribution of investigational drugs by testing phase and success or failure outcome. We then can calculate weighted average clinical phase costs. And this gives us phase costs per investigational compound, per drug that enters the clinical testing pipeline. Now, there remains the problem of getting a pre-human or out-of-pocket cost estimate. That estimate is linked to the project-level clinical period cost estimate. It's the aggregate company time series R&D expenditure data that I mentioned earlier that provide the link between the two estimates. The link recognizes that there is a lag between when pre-human expenditures are made on average and when associated clinical expenditures are incurred on average. Now, as I mentioned, we need estimates of clinical phase transition rates and approval success rates to get expected costs and translate costs per investigational drug to cost per approved drug. For that, we went to the population level and analyzed data in a data set that we have of the top 50 pharmaceutical firms on drugs and biologics that met our study inclusion criteria. Those were analyses that I did separate from those on the cost survey data. That allowed us to use to input results from a larger data set. However, I also estimated success rates for just the 10 participating firms, and the results were very close to what I got for the larger set of firms. There was less than a 1 percentage point difference in the overall clinical approval success rate. Now, we used the survey information on when the compounds were synthesized, when human testing first began, when particular clinical phases began, and when those clinical phases ended to get estimates of average phase lengths and phase overlaps to get a representative time profile over which we capitalize costs. The regulatory review phase data were taken from FDA data on marketing application submission and approval dates for all new drug approvals. Now, we capitalized all costs to the point of marketing approval. The approval to which we keyed the timing was U.S. approval. The discount rate, or interest rate, if you will, that we used was an estimate of the weighted average pharmaceutical company cost of capital over a relevant period. The methodological approach used to get company cost of capital was the application of what is called the capital asset pricing model, a commonly used method in finance to assess the rate of returns investors expect to get them to invest in a company. I should say, as we get to the results now, I should say that all costs in the study have been inflation adjusted and are expressed in year 2013 dollars. These costs include costs for everything R&D related during discovery and development, and include all indications pursued up to the point of marketing approval for the pre-approval cost estimate. Now, we can't use the mean phase cost results for investigational drugs and simply add them up, because not every drug that enters the clinical testing pipeline will make it to late-stage clinical testing. So we have to get expected costs, and that means finding estimates of what are called phase transition and clinical approval success rates. This slide just informs you of the data set that was used to get these results. The results are based on information through the end of 2013 on 1,442 compounds that met study inclusion criteria. We used a variety of publicly available data sources to construct the data set. Only 7.1% of those compounds were approved, and the vast majority, 80.3%, had been abandoned. But 12.6% were still in active development at the time of analysis, and you can expect that some of those will eventually be approved, particularly since they tended to be in late-stage clinical testing. And our methodology takes that into account. Now, the results of our success rate analysis is given here. So this says, for example, that only about 36% of the drugs proceeded from phase 2 to phase 3 testing. The other 64% that had undergone phase 2 testing failed at that phase. For drugs that underwent expensive phase 3 testing, only 62% had an application submitted to the FDA for marketing approval. And then there's a certain failure rate even after you submit an application to the FDA for marketing approval. So 38% of compounds actually failed in phase 3 testing without even having an application submitted to the FDA. Now, the overall clinical approval success rate is the last bar in the graph, and it is the likelihood that a compound that enters the clinical testing pipeline will eventually be approved for marketing. It's the product of the percentages of the bars to the left of it. So, and that estimate is a little under 12%. So approximately 7 out of 8 compounds that enter the clinical testing pipeline will fail in development. Put another way, this says more precisely that on average you need to put 8.5 compounds in clinical development to get one approval. The overall clinical approval success rate that is relevant to this study here, the 11.83%, is actually much lower than it was for the previous study. And I will show later how importantly that impacts our results. Yes.
Can I ask a quick question?
Sure.
You said earlier that there was 106 investigational drugs were like that, but then you said the universe of compounds you studied were 1442. Well, how do those two figures compare? Are you studying 1442 compounds, or are you studying 106?
For the success rate analysis, which is an input to getting the overall cost estimate, we use the 1,442. Right.
But for the overall cost, you use the 106.
Yes.
Okay.
Now, to get time costs and capitalized cost estimates, we need, as I said, estimates of development and approval phase times. We need to look at overlaps between phases. The main development periods, though, are phase-to-phase times. That is the time from the start of one phase to the start of the next. So we have those average times for the whole process here. The entire period from the start of clinical testing to marketing approval was a little more than 8 years. The time from synthesis to approval was a little under 11 years. The synthesis to human testing period was 2.5 years, which is actually down from 4 years in the past. And these results are used to develop the time profile over which costs are capitalized to the point of marketing approval. Now, we need to also take care of the pre-human side of the process. Our development time estimates suggest approximately a 5-year lag between the median of the period when pre-human expenditures are incurred and the median of the period when clinical testing expenditures are incurred. With no lag imposed on the data, we found that the average annual pre-human expenditures are 38% of the total R&D expenditures. But with the 5-year lag imposed, that share is just under 31%. And that is very close to the figure that we had for the last study, which was 30%. We can use that percentage along with the compound level clinical period cost estimate to deduce what the pre-human cost per approved new drug should be. But the results are actually not sensitive to different assumptions about the lag, at least within reason. They change very little, for example, if we use a 4-year lag or a 6-year lag. So by now in this process, we would have estimates of the cash outlays for investigational drugs by phase, phase transition and clinical approval success rates, and development times by phase. What we need to complete the picture and get a full cost estimate per approved new drug that includes not only the cost of failures, but also the time costs of development is, in effect, an interest rate at which to capitalize the cash outlays. And that interest rate is a weighted average company cost of capital for a relevant period. The weighting consists of sort of looking at returns for both debt and equity, although funding for these firms is overwhelmingly equity-based. And we want to get a real, that is, inflation-adjusted estimate. So here you see we looked at various years that would cover development during the period we analyzed. The capital asset pricing model was applied to public data on publicly traded biopharmaceutical firms. We settled on a base case rate of 10.5% to capitalized cost. But you can do sensitivity analysis that gives you what the results would be using other values for the cost of capital. And we do that in a paper as well as look at the effects of varying other estimates. And some of that analysis will be shown later. Now, we can finally get to the top line results. Here we have costs on both an out-of-pocket and in a capitalized cost basis, and by pre-human, in clinical periods, and in total. So in terms of cash outlays, total cost per approved new drug is about $1.4 billion, with 69% of that coming during the clinical testing period. That, I should point out, includes the costs of failures. If we add time costs to get the full capitalized cost estimates, we get about $2.6 billion per approved drug. And because pre-human expenditures occur further back in time than do clinical expenditures, on a capitalized basis, pre-human expenditures take a larger share of the total. That is, the time costs for those expenditures are relatively higher. So the clinical share of the total thus drops from 69% to 57%. In total, time costs account for 45% of the total. That is down from about 50% in prior studies. Now, we were also able, as we did for the last study, to use both survey data on aggregate annual company R&D cost breakdowns and external data to estimate post-approval R&D. That is, we estimated expenditures on R&D conducted after original marketing approval. That would include R&D on new indications, new formulations, regulatory required post-approval commitments, new dosage regimens, and new dosage strengths. The estimated cash outlay per approved drug for post-approval R&D was $446 million, or 25% of total life cycle R&D out-of-pocket costs. Now, when you look at capitalized costs, you have to capitalize everything to the same point in time. And the time mark that we have been using is the date of marketing approval. Post-approval R&D costs occur in the future relative to that time point, so they have to be discounted back to that point. That means that the capitalized cost for post-approval R&D will be less than the out-of-pocket costs. So the capitalized cost of post-approval R&D is $312 million, and that is 11% of the total life cycle capitalized cost per approved drug, and that total is $2.87 billion. Now, with respect to pre-approval costs per approved drug, we now have a series of publications with similar methodologies. And if you pay careful attention to the periods analyzed in the 3 prior published studies and in the current study, you can compare costs by period. In constant dollars, total capitalized R&D cost per approved new drug rose from $179 million for the 1970s to nearly $2.6 billion for the 2000s to early 2010s. That's a 14-fold increase, but it's more instructive to look specifically at annual growth rates between the studies. Now, noting the periods analyzed in the 3 prior published studies and in the current study, you can estimate compound annual growth rates between the studies, and so between periods. I looked at this in terms of when approvals would occur for the development analyzed in the various studies. So, roughly speaking, the results for the first 2 studies can give you a growth rate in R&D costs between 1970s approvals and 1980s approvals. The second and third studies can be used to give you growth rates in R&D costs between the 1980s and the 1990s approvals. And the last study and the current study can give you growth rates in R&D costs from the 1990s up through to the early 2010s. So this slide shows those growth rates for out-of-pocket costs. In terms of total out-of-pocket costs, which include failure costs, expenditures per approval grew at higher rates over time. It's 9.3% per year for the current study period relative to the study period for the prior study. There are differences by testing phase. Clinical period costs grew at a very high rate over the prior study period. That growth has declined somewhat for the current study, but it is still high. The growth rate for pre-human testing fell significantly for the last study, but those costs have risen substantially for the current study. The growth rate for pre-human testing for the current study period was similar to that for clinical testing. Now, the results on the previous slide account for changes in average cash outlays and the proportion of drugs that fail in testing. We can add time costs here and look at growth rates on capitalized costs. Here, because the development timeline became somewhat shorter and the cost of capital fell, you don't see the upward trend in total cost that you saw for out-of-pocket costs. The compound annual growth rate for total capitalized costs for the current study relative to the previous one is 8.5%, and that is less than the growth rate between the 1970s and the 1980s. The patterns of change at the pre-human and clinical levels, though, were qualitatively similar. Okay. But let's see if we can take a closer look at what elements of the process seem to be driving the increase in costs that we found. So, I examined for the prior study and the current study the extent to which various components of the drug development process, cash outlays, development risk, development time, and the cost of capital contributed to the increase in total R&D costs. You can't completely isolate the cost impacts of changes in individual parameters or estimates because they interact with each other. The effects are not linear or otherwise separable. However, you can still get a good sense for what is important by looking at individual changes. The increase in total capitalized costs for this study relative to the prior one in constant dollars is 145%. And with an approximate 11-year difference between the study periods. Now, I can account for 124% of that by looking at individual changes. The rest you can attribute to, if you will, interaction effects. So I divided this analysis into 3 slides. The thought experiment here is to suppose that all estimates for the previous study are held constant, but you change one estimate to what its value is in the current study. So the base here is what we found for the previous study, which was 1,044 million, 1.044 billion in year 2013 dollars. So in this slide, I am looking at changes between the studies in average cash outlays for investigational drugs for a development phase. Specifically, we're not considering changes in failure rates. We are holding that and all other estimates constant. And we can see that the increase in average cash outlays for conducting R&D during development accounts for a large share of the increase in total costs. This effect can result, for example, from increases in clinical trial complexity, clinical trial sizes, and per patient costs, both of which evidence in other studies, including some done at the center, indicate have increased over time. Okay. Now, what may not be well appreciated, though, is the effect of a shift in the risk profile for clinical development. There was a substantial decrease in the clinical approval success rate between the studies. It fell from 21.5% to 11.8%. There was also a shift in the distribution of failures across phases. But if we hold that distribution constant and just look at the effect of the change in the clinical approval success rate, we get a 57% increase in costs, just from that factor alone. There was some movement toward feeling the failures faster, and that has a moderating effect on cost. Here you can see that effect considered alone would account for a 6% drop in costs. If we take everything that happened to the risk profile into account, changes in the failure distribution and changes in the overall clinical approval success rate, you get a 47% increase in cost. Finally, we can look at the impact of changes between the studies in the time profile for development and in the cost of capital. Here the effects are modest. The clinical development period was somewhat longer, and that has a small positive or increasing effect on costs. However, that effect was overwhelmed by shorter pre-human and regulatory review periods. In aggregate, changes in the time profile had a modest moderating effect on costs. Finally, the change in the discount rate used from 11% for the previous study to 10.5% had a minor moderating effect on the increase in costs. So in sum, we found that R&D costs grew significantly since we last measured this. The fully loaded cost per approved new drug, inclusive of time costs, increased at a compound annual rate of 8.5%. Considering only the cash outlays, inclusive of the cost of failures, cost per approved new drug grew at a 9.3% annual rate. We found in particular that the clinical approval success rate fell significantly. The higher failure rate had a substantial impact on R&D costs. Higher out-of-pocket costs of conducting R&D and proportionally more failures in clinical testing are what really drove the increase in cost per approved new drug. Finally, while time costs remained significant, a somewhat shorter total discovery to approval period had the effect of moderating the rate of increase in R&D costs. A somewhat lower cost of obtaining capital also had a modest impact on holding down increases in the economic cost of new drug development. Changes in development times and the cost of capital together reduced the share of total economic cost accounted for by time, by what we're calling time costs. So thank you, and we can open it up for questions now.
Joe, thank you for a very illuminating talk, a very struggling often to try to understand, but I think you made it as clear as you can. I had actually 3 questions. One had to do with the cash outlays for preclinical development and how much of that reflects outsourcing as contrasted to doing it with in-house, which increased over that decade.
Okay, the question was, I think, related to preclinical, and the question was, to what extent does outsourcing occur over time, or was a factor in?
It costs more to go out to somebody else than when these companies had the capacity to do this work in-house, which fewer of them have these days.
Yeah. Well, unfortunately, we don't have the data on that to see if there was a distinction, and we don't have the data to do that for the clinical period too. We don't have the data in this study to differentiate between clinical costs incurred internally versus clinical costs that were outsourced. There are a number of studies, as you may know, that the center has done on the CRO sector and their impact. And Ken Getz is here, and perhaps he can talk to you a little bit more about that. Sure. But for this particular sample, we don't have that kind of breakdown.
And the other is that within the 100 and some odd drugs that you actually focused in on, by therapeutic class, is there a big range in cost of development?
Okay. The question is basically the costs vary by therapeutic class. We certainly found in the past that's the case. We're going to follow this analysis up with a more in-depth analysis on costs by therapeutic class. There's some in the paper. There's some analysis along those lines. We found some differences in clinical phase costs by therapeutic category. But we'll do a full bore analysis as we did for the overall estimates in a follow-up study for drugs by therapeutic class. We limited in sample sizes by class, so we'd have to restrict our analysis to fairly broad therapeutic categories, and we had done that in the past and found differences in costs in clinical period costs, which is all that we could measure from the data.
Thank you.
Ken?
Joe, thank you very much for this presentation. I was struck by the growth rate, the annual growth rate in the capitalized costs, and how reasonable it seemed relative to even the growth rates we've seen in overall R&D spending. And I was wondering if you could comment on the reasonableness of that growth rate.
Okay. Reasonableness. Well, there actually, we made a lot of effort in the study, in the paper, to validate our results, if you will. So we did a number of analyses that looked at components of the process and focus actually a lot on the growth rate estimates that we found in the data. And we did find that we could certainly support those growth rates when you look at a variety of measures and put them together from outside sources, put them together in ways that would suggest what the growth rates might be. That can include data that's out there on cost per patient, clinical trial complexity, number of subjects in trials, data on salaries of scientists, changes in employment levels at pharmaceutical firms and among CROs. So there's a lot of that in the paper that quantifies, if you will, the reasonableness of the growth rates and costs.
It seems like a lot of the drug approvals that I've written about are on drugs that were licensed by a pharma company from a biotech. How often does that happen, and compared to situations where a pharma company develops a compound in its own lab and takes it all the way through to commercial approval? And how do those two different models vary in terms of cost, if at all?
Okay, well, we actually did a study recently, Ken Getz and I and someone else from the center, that looked at drug approvals during the 2000s and sort of classified drugs according to whether they were developed entirely by a single firm all the way through, or there was some kind of business deal involved, during the process, whether it be licensing or co-development, some acquisition, and approximately about half the approvals went all the way through under the auspices of a single firm. I don't remember the exact percentage for licensing drugs, but that was somewhere in the 20s, something like that, 20% range. In terms of the costs, we don't know that. We don't have a full set of data to indicate whether there are, what differentiations there are, if any. We can point to certain evidence for aspects of the process. We can hypothesize. There's nothing sort of inherently special about licensing drugs, relative drugs developed entirely by a single firm in regard to testing and testing requirements. The FDA, other regulatory authorities aren't going to give a break, if you will, to a compound in testing just because it happens to have been licensed. So for that reason, that there wouldn't be any reason to expect any differences in cash outlays. We did, however, there may be one reason why time costs and in particular and perhaps even cash outlays may be somewhat higher for licensing products, because that study that I just mentioned, where we looked at, we classified approvals according to whether or not there was some sort of business arrangement or business deal during development. We did find that the clinical development period was somewhat longer for compounds that had some sort of arrangement like this, were licensed in or in the co-development, that sort of thing. Perhaps that reflects operational disruptions when there's a handoff of a drug from one company to another. That alone would suggest somewhat higher time costs, perhaps even higher out-of-pocket costs. There really isn't evidence that for higher success rates for clinical drugs, when we consider the population from which they come. You really have to know. Basically, it's coming from, if you will, primarily from the small firm sector. So you really have to know something about success rates for the small firm sector. There's some evidence, some limited evidence out there that suggests that actually the success rates could be somewhat lower for small firms. There's a number of studies where there's evidence of that, either completely or partially. There's also reason to believe that small firms will tend to keep their drugs in development longer than larger firms might, because basically they're single or two-product firms, and if the product fails, the company goes under. So there's actually some evidence that small firms push drugs from early phases into mid to late stage clinical testing more frequently than do larger firms, but then fail at higher rates later. So, which actually also contribute to higher costs for the smaller firms. It's also the case that there's been a fair amount of research that indicates that costs of capital are higher for smaller firms, and this is all getting back to the licensed drug issue, because you really have to look at not just the licensed drugs, but the population from which those drugs are accompanied, the universe of compounds where those drugs where firms are looking, if you will, to license out their drugs.
I know that the ultimate drug prices weren't taken into account in this study, but just seeing as this is often used as kind of justification for those prices, do you have any sense of the relationship between the increase in how much it costs to develop a drug versus the increase in what the drugs cost at the end?
Well, if you look at it's sort of the individual product level and the pricing in an individual product, it's not going to be determined by the prior R&D costs for developing that drug. It's not going to be determined based on an average industry R&D costs. The prior costs are sunk costs. They're, firms going to look at the market, the competitive landscape, what reimbursement authorities require of them, what reimbursement systems are like to determine the actual price. But what we can say is that R&D costs are an important component of the profitability of new drug development, and so are important in assessing the incentives for innovation. So, for example, if R&D costs are increasing over time, but reimbursement levels in general remain the same, you'd expect less drug development. You expect fewer products to appear to be financially viable, and so eventually get less innovation in the future. Conversely, if costs remain the same but reimbursement levels declined, then you expect the same sort of thing. So it's part of the mosaic that influences incentives for innovation, and so ultimately innovation in the future.
I have some questions that have come in from those listening online. This is from the pink sheet. Is a breakout figure available for the amount spent on drug firms testing comparator drugs to accommodate payer demands for comparative effectiveness data? This is mentioned in the press release as one of the factors that likely boosted out-of-pocket clinical costs.
Well, we think we're seeing more of that, and so and there's every reason to think that would has a positive or an increasing effect on costs. But fortunately, we don't. The data for this study wouldn't allow you to make that distinction. So we can't tell how much comparator testing was done on these compounds, and how much was just sort of placebo-control based.
Okay, this question is from BioWorld Today, and it's multi-part, and a lot of it has to do with separating out different classes of compounds. So the first part, did the study separate development costs for small molecule drugs from those of biologics, or can you provide any breakdowns?
Okay, there is a breakdown in the fuller study. You're limited to some extent by the numbers here. What we found by looking at costs at the individual phase level, that doing a statistical analysis, a statistical regression analysis, we could not find differences between large molecules and small molecules for phase 1 and phase 3 costs, but we did find higher costs for the large molecules for phase 2 testing, which I would note includes all indications pursued would include all indications pursued during phase 2. So it may be that more is done, more indications are pursued for the large molecules. And I should say that this is controlling for differences in the therapeutic class distributions across the large molecules and small molecules, because there is a difference in the therapeutic class distribution between large molecules and small molecules. That's cost per investigational drug at individual phases. Ultimately, you're interested in cost per approved drug, and it's the case that our analyses show that costs per approved compound for large molecules are significantly higher than for small molecules. And once you take that into account, the total clinical period costs per approved molecule are actually lower for the large molecules, about 45% lower, which is actually remarkably close to the percentage difference that we found in a study that we had published in 2007 that looked specifically at a a cost of, quote unquote, biotech compounds, large molecules, recombinant proteins, and monoclonal antibodies. So, some evidence of higher costs, at least at the phase level for investigational drug level for phase 2. But once you take into account the higher success rates for the large molecules, you actually get lower clinical costs. And in that 2007 study, we found, though, that there were higher pre-human, discovery and preclinical development costs for the large molecules relative to the small molecules, so that actually in the end, the higher discovery and preclinical development costs and the lower clinical development costs essentially evened out, so that the overall cost estimates were essentially the same.
Okay, the next question: how many, if any, of the drugs in your sample received orphan designation?
You can't know that because the vast majority of the drugs were investigational drugs, or at least I don't know that. You can look certainly at the approvals in the sample, and I believe there was 11-12% had received an orphan drug designation. Now, there are lots of drugs in investigational drugs that, we don't know whether or not they hadn't gotten orphan drug designation. They may not have yet gotten orphan drug designation before they failed. So it's hard to say, since we're looking at investigational drugs as well as approved drugs, and a very large share of the sample consists of investigational drugs. But I'd say that we have every reason to think, through other analyses, that the distribution of compounds by therapeutic class and by phase outcome is representative of the larger group from which the survey data were obtained. So it's in all likelihood representative of the share of drugs that have received orphan drug designations for some indication or indications for that set of firms. It's probably true that the orphan drug development is more concentrated in the small firm sector.
Okay, and the last part is, even though the goal was to determine an average, could there be a huge range depending on the complexity of the drug and whether it's first in class and the therapeutic area? But you've already talked about the therapeutic area. So just something about the first in class.
It's possible. It's difficult to make that distinction. What's usually called a first-in-class drug is a drug that makes it to the market first. But what we found from other studies is that if you look at the development histories of these so-called first-in-class drugs based on approval and follow-on drugs, which are also often derisively referred to as Me Too drugs, if you look at their development histories, they were more or less being developed at essentially the same time. So it's just that one drug happened to win the race and has been labeled the first in class. What's often going on here is that the science is driving firms, a number of different firms, into the same area. So they're looking at the same pharmacologic class at the same point in time. So that makes it difficult to make any sort of useful distinctions, I think, between first in class and other drugs.
Count all the global costs, but they run clinical trials in different countries with different requirements, and of course they have to justify the cost to these payment committees and panels in Europe and everything. Is all that factored in?
It's all factored in. These are global costs up to the point of U.S. marketing approval.
Of the U.S. market.
Right. And then the post-approval R&D estimate covers everything that happens after that. But it's global expenditures.
Given your significant increase from your last study you're seeing now, innovative drug, can you speculate what impact that might have on biosimilars? I mean, you're not talking about innovative drugs, but the overall cost of developing those type of drugs is a significant risk factor. It's not necessarily a sure thing. But seeing a significant increase in innovative drugs as you're reporting now, do you have any speculation how that might impact the biosimilar area?
Well, I hesitate to say without data. I'm actually working with some people at MIT on looking at the economics of biosimilar development versus novel biologics versus biobetters versus small molecules, and so we're hoping to get at some answers to. With respect to the overall economic rates of return to those various types of investment. But that's sort of something that's in process now.
It's all about economics.
Right.
It's critical.
Okay, it's 11 o'clock now, so we are going to end this this discussion. Thank all of you. I thank all of you on behalf of the center and the authors of the cost study for attending, as well as those that are online. Feel free for the next hour to discuss any specific questions you might have with Joe. But going forward, feel free to contact the Tufts Center, and we will try to answer your questions. So thank you again.